speaker before we have a break for morning tea is Pauline Cervantes, and Pauline has brought an amazing amount of culturing experience to us from all this knowledge she stole from the endophyte group up at Lincoln Uni. So we're so privileged to have her down here studying the fungal endomicrobiome of Kinocloa.
Hi, good morning, everyone. So I'll be taking you to the other side now to look at ascomycete fungi. And so my name is Pauline Cervantes. I am a PhD candidate at the botany department in the University of Otago. And so this morning, I'm supervised by associate professors Paul Guy and David Orlovich. And this morning, I'm going to be giving a brief overview of my what I'm doing for my PhD, which is looking at the fungal endophytic microbiome of New Zealand's native tussock grass, Kinocloa, and in particular, Kinocloa rigida, as well as Kinocloa rubra. So what exactly are endophytes? So endophytes is a term used which includes all microorganisms, from archaea to bacteria to fungi, which inhabit the inter or intracellular spaces of plants, whether if it's for part or all of their life cycle, and they don't appear to have any damage to the host. So most fungal endophytes, which is what I'm interested in, are belonging to the phylum Ascomycota, and they're typically grouped into two. So this is the systemic endophytes and non-systemic endophytes. And they differ mostly in the way that they're transmitted, the extent in which they colonize their host, as well as the extent of the effects that they have on their hosts. So when we look at systemic endophytes, it often refers to fungi in the family Clavicipitaceae, which includes the well-known epicloe endophyte that is often associated with pasture grass systems. So systemic endophytes like epicloe are able to systemically colonize the plant, the above ground tissues of the plant all throughout, as you can see on this image here. And their hyphae is also able to grow into the seeds, so then they're able to be passed on to the progeny and be vertically transmitted. And their effects on their hosts are very well known. They produce secondary metabolites, which helps protect the plant against biotic stress, such as damage from any insect pests. And when we look at the flip side, the non-systemic endophytes, they're thought to be, I guess, the opposite, where they involve a wider group of endophytic species or fungal species, and they're thought to be horizontally transmitted from fungal inoculum, whether it's from air or the rain or the wider soil microbial pool. And the extent of the colonization in their host is usually much more distinct from epicloe in a way that it's more localized and restricted. So these images are examples of dark septate endophytes, which have colonized the roots of a plant. And so when we look at their effect on the hosts, it's not as well known just because of their wide host range. And when we think about endophytes as a whole, it's better to think of their interactions along the mutualism to parasitism continuum, because at any point in time, they can also become pathogenic when the host plant is stressed or when resources become limited. And when we look at endophyte research here in New Zealand in particular, there's a lot of studies that's being done looking at foliar fungal diversities, with some examples over here. And in particular, there's more recent interest in looking at native medicinal plants, in particular in myrtaceous species such as Kanuka, Manuka, and Ramarama, and Purukawa. And recently, this report that I read by Ridgway et al., they were looking into endophytes or native endophytes in these species and seeing how these native endophytes could be used potentially as a biological control agent against the introduced pathogen myrtle rust, which I thought was really, really interesting. And when we look at native grasses in particular, there hasn't been a lot of research looking at endophytes, or particularly non-systemic endophytes, and it's mostly being focused on seeing whether or not these species could be hosts to epicloe, and that's for good reason, because the benefits that epicloe does provide is very good for the host if it's present there. So what I'm doing for my project is sort of a continuation of my honors project back in 2020, which was looking at endophytes, the culturable endophytes in Kainakloa. So that, as well as the findings that I've gathered so far, will hopefully work towards identifying the endophytic microbiome of Kainakloa, and in particular, Kainakloa rubra and Kainakloa rigida. So how I've done that is I've collected at least 100 plants from 10 different sites with different land use histories. And I've also collected 15 soil core samples within each site, because I want to later on look at physiochemical factors within the soil to see whether or not some of these factors like pH and carbon and nitrogen can be used to help us understand what the assembly of fungi within Kainakloa. So all in all, for my project, I'll be using a culture-based and culture-independent approach. So for now, in the last 6 to 8 months, I've been focused more so on the culture-based approach, and the results that I'll be talking about later on will be from that. So once I've gathered the plants, I then cleaned and surface sterilized them using the sequence of Trident X, ethanol, bleach, as well as cleansing it with sterile RO. And I looked into the blade, sheath, and pseudostem region, as well as the roots. And if the plants were flowering, I also took the spikelets to give some sort of indication on whether or not some of these endophytes could potentially be vertically transmitted. And I also took some controls before and after the surface sterilization process to see whether or not the surface sterilization worked. And usually after about 2 weeks, this is what the plates look like here below. So in some cases, you can have as much as 16 different species of endophytes in one plate to as little as 1 species in a plate. So these 16 endophytes is usually a hard day for me, because it's a lot of trying not to contaminate the plate, which is this plate is very much easier to do. So usually after 2 weeks, when the pure colonies have reached good growth, I then morphotype them based on their sites and grouping the similar looking ones as one particular morphotype. And then I later on take a subset of each morphotype for molecular analysis, and then extract the DNA, amplify that, and with Sanger sequencing, I can get the ITS region and compare that to existing databases like Unite and GeneBank to identify them. And I've been also looking at their microscopic structures, if they're sporulating, as well as their hyphal characteristics, to have multiple lines of evidence to prove and to justify the IDs that I've associated with the colonies. So what this figure shows is it represents the percentage of fungal colonies that have emerged relative to the number of total tissue segments for both Kainakloa rigida and Kainakloa rubra. So there appears to be more viable fungal propagules in Kainakloa rubra. However, this does not necessarily reflect species diversity, because when we look at this particular example here, once again, 16 CFUs can represent 16 different species, whereas if you look here, 20 CFUs, it only actually represents 1 species. So in order to actually assess the core taxa in Kainakloa, I looked at frequently occurring morphotypes and quantified how frequently they occurred within a particular site, as well as between populations in different sites. And so after plating about 8,000 tissue segments later and 3,000 subcultures, I was able to identify what I refer to as the core endomicrobiome, and which refers to, I guess, key taxa that occurred frequently within and between sites. And 3 of the 10 sites that I looked at, which I sampled in 2020, I also sampled back in 2020. So I was able to confirm some level of consistency over time for some of these core endophytes. And for the ones that appeared in the spikelets or in the flowering parts of the plant, it provides some evidence that some of them may be vertically transmitted. So far I've identified 11 key morphotypes, so some of which occur in between 10 to 20% of plants sampled. So that includes taxa like Fusarium, Cladosporium, this Albifimbria verrucaria-like colony, and mucor. And in some plants, in some cases, they can appear to as much as 80% of the plant sampled. So that's like Arthrinium and Phylocephala. So I'll be basically going into more detail into the morphotypes with higher isolation frequencies and exploring what is present in the literature about endophytic strains within this taxa. So let's just take a moment to appreciate the beautiful ascomycete fungi and all how colorful they are, and don't they look beautiful? So in Arthrinium, it was initially observed back in 2020 from 3 different sites that I looked at, and this year I observed them again from those same sites in Kinochlora rubra and Kinochlora rigida, and they were present as well in the new sites that I looked at. So all in all, I isolated them from 79.7% of plants across all tissue types and twice from the flowering parts. So Arthrinium is known to have species which are plant pathogens, saprophytes, as well as endophytes. They're more commonly isolated in terrestrial, but also in marine environments, and their biological activities have also been explored in the literature, and it's been used in vitro as a biological control agent against one particular aquatic plant pathogen. And in 2020, I also used Arthrinium, a strain of Arthrinium, as a biological control agent against Botrytis cinerea, which showed some success. And the next morphotype is Trichoderma, and I initially isolated this from the root tissues of Kinochlora rigida in Swampy Summit back in 2020. And more recently, I observed this again in both Kinochlora rubra and Kinochlora rigida. They're both present in the roots from 52.7% of plants that I looked at. So they were also present in all the 7 different sites. So Trichoderma is a very well-studied soil-borne fungi, and they're known to colonize plants as a root endophyte, and they've been applied as some strains have been applied as a biological control agent in many scenarios. And one example, it's actually been commercialized here in New Zealand, Kiwivax, which has been developed in Lincoln University, has been used as a control agent against PSA in kiwi fruit. And it's also capable of parasitizing hyphae in fungal pathogens like botrytis, whilst also promoting plant growth. So another core group is Penicillium. So back in 2020, I didn't find any Penicillium, but this time around 70.3% of plants hosted at least one species of Penicillium, and there's a huge diversity that I was able to isolate, as you can see from the colonies here on the right. However, there was one particular colony morphology which I observed more consistently within plants of each site and between sites as well. And they were also observed across all tissue types. And so Penicillium is a very common fungi, if not probably one of the most well-known fungi. They're present basically almost everywhere, and they have a swathe of very diverse biological activities. Some strains are known to have anti-cancer properties, and more recently, Penicillium is being explored as a source for fungal pigments as well, because as you can see, they produce all sorts of pigments in vitro. So another group is the dark septate endophytes, which was isolated from 85.14% of plants. And this group represents at least 3 different species. And I was able to isolate dark septate endophytes across all tissue types, and Phylocephala, which is this particular one, was more so dominant in the spikelets and the roots, and they were also observed in all sites. So this group is often characterized by their ability to produce microsclerotia in the root tissue of their host plant, and they also have melanized hyphae, as you can see in the pictures here below, the dark brown pigmentation. And it's this pigmentation that's being associated at times for their ability to protect their host against abiotic stress, such as salinity, as well as drought. And another key group that I'm interested in and was able to isolate were some species of entomopathogenic endophytes, which includes Metarhizium as well as Bovaria. So I ID'd these based off of their colony morphology as well as their spore structures. So these particular endophytes are very ideal when it comes to use as biological control agents, in particular against insect pests, because of their ability to establish in their host plants. So when you compare these to using insecticides, their non-target impacts are much lower. So looking at how these endophytes can be applied outside of their host Pinocloa, I then want to continue looking at Arthenium, Trichoderma, Penicillium, Phylocephala, and the entomopathogenic endophytes, and assess in vitro what their potential ecological functions might be. And I'm able to do that because I have the cultures on hand. And some of the things that I also want to do with them is test them against fungal pathogens. Like in 2020, I tested Trichoderma, a strain of Trichoderma and Arthenium that I isolated against Botrytis cinerea, and it showed some success, and I want to add more endophytes onto that list. And currently I've inoculated onion seeds as well with Metarhizium, which is a project that I'm doing for and has been funded by Onions NZ, and I want to assess whether or not the endophytes are taken up by the plant and whether or not it could potentially have some sort of effect on their growth. And if it establishes well into the plant, it could have some implications in protecting the plant against insect pests, but that's still ongoing. So basically, in summary, there appears to be a difference in overall colony forming units in both Kinocloa rigida and Kinocloa rubra. So this reflects the fact that there's more viable fungal propagules in Kinocloa rubra. But when we look at this in terms of key morphotypes that are present, the core endophytic genera is consistent between both species, regardless of even between the different sites. So it is likely that these microbes are probably coming from the wider soil microbial pool, and potentially there's a mechanism that these plants are able to attract these particular endophytes. And in some examples where the colonization is systemic and I've been able to isolate them in the flowering parts like Phylocephala and Arthenium, it suggests that these endophytes could also be potentially vertically transmitted, and the fact that their colonization is very extensive and that they could be potentially vertically transmitted suggests that they have a wider effect, I guess, to their host. And it is these taxa that I want to continue on to look at in terms of their biological activities. And it'll be really interesting to see as well, once I've done the culture independent side of things, whether similar taxa pops up. So I would like to say thank you to my supervisors, Paul and David, and to everyone who's helped me out in my project. And this list will probably continue to grow. And questions?
Fantastic. Thank you, Pauline. We've had one comment about the beautiful images from Kath there. It's mind-blowing, really. Peter J had a question. Peter, were you able to ask that yourself in real life?
Possibly. Can you hear me?
Yep. Yes, we can.
I just wondered if you're finding any epichloroid-like fungi in these grasses.
It's hard to say because I actually haven't been, besides looking at what's been published in the literature, they appear to, they look very cryptic, and there's a lot of like white sort of colonies that I find, and it's hard to know for sure what exactly they are until I get the DNA barcoding data. And I've been really focused on morphotyping them. So then I won't get that until the next couple of months. And there could be some, but because I have thousands of cultures that I sort of have to go through. I'm not 100% sure whether or not epicloe is present there, but with the surveys that with what has been published so far, there's no study that's said that epicloe has been hosted by any species of Kinocloa. But it'd be interesting to see if any of the colonies that I do end up subculturing, if they are. But it costs a lot of money as well, and I have quite a lot of colonies, so it's a matter of prioritizing which ones I think are important, and that's been a big task too as well. Yeah.
Yeah, so talks I've listened to from the Palmerston North guys in the past, well, years and years ago now, they seem to be quite distinctive morphologically inside the plants. So it might be worth just, kind of having a look inside.
Yeah, because so I think hopefully later on in my project too, my supervisor Paul wants to look at sort of transects of the tissue under electron microscopy. So hopefully that could show us something, whether or not there is epicloe there. And it'll be also interesting to see how endophytes are distributed within Kinocloa, within the tissue as well.
Great. Okay. Any more questions? No, I think that, here we are, Bevan. Any intent to describe these as new species? Oh, that's a good idea.
Oh, yeah, that would be cool to do. I'd be very keen to do that, but in terms of how much time I have doing my PhD, I don't think I'll be able to do that. So I've been sort of focused on looking at how like in vitro assays and looking more so on their functions rather than spending a lot of time in identifying them down to the species or strain level, because even when I've gotten ITS sequences back, sometimes what comes up in UNITE and GeneBank often contradict each other, and the identities are sort of it's hard to be super sure when it comes to their IDs. That's why I've been also looking at their microscopy stuff, but that would be cool to do if ever in the near future, hopefully. Yeah.
Yep, maybe we could get another student onto that, or 25 more students onto all of those. Okay, that's great. And Paul Guy has just responded, I guess, to Peter Johnston's comment there, saying that epicloe only found so far in povoid grasses. Yep. Right. Okay. Well, look, gosh, what a great start to the day. I think it's just such a fun and exciting day.